just minions.
I'm Hongshi.
I'm Junher.
I'm Danya.
I'm Lei.
And I'm Shannon.
Today, we're going to be talking about
enzymes and digestion.
Have you ever wondered what happens to your food after we eat?
Did you even know that there are enzymes in your body to help you digest food?
what even are enzymes?
And how do they even work?
And why do we need them?
Guess what? We're going to tell you all about it.
Before we get any further with enzymes, we have to understand the concept of digestion. Digestion is categorized as mechanical and chemical. Mechanical digestion refers to the breaking down of food through physical processes such as the chewing of the mouth and churning in the stomach to make it into smaller pieces. However, this digestion alone is not enough because these food molecules are still water insoluble and could take weeks to be absorbed into our body. The purpose of this process is only to increase surface area for chemical digestion.
On the other hand, chemical digestion plays the role of breaking down food from insoluble molecules into small water-soluble molecules with the help of enzymes. This helps with absorption of nutrients into our bloodstream. Enzymes are defined as biological catalysts that speed up chemical reactions. This basically means that they don't experience changes during the breaking down of food molecules, but are present to expedite the process.
These enzymes have active shapes that are complementary to the particular substrates. For instance, after we eat a burger, the enzymes in the stomach will degrade the protein found in the burger. This process occurs as the enzymes attach to the protein molecules and only the protein molecules because they have shapes that fit together. In this example, the substrate is the protein, and as the enzyme and substrate combine, they are called the enzyme-substrate complex.
Due to the lock and key nature of the enzymes and the substrate, they are unable to break down any other substances besides the one they were built for. This is why lactose-intolerant people cannot process dairy products. They have a lack of the specific lactase enzymes to break down lactose, making them unable to absorb lactose sugars into simple sugars. Therefore, the unabsorbed food will be released through egestion, which means defecation.
Sometimes the enzymes are inactive and are required to first bind with a non-protein substance called a cofactor to activate it. Usually, these cofactors are made with vitamins.
When a specific enzyme's activity is higher than our bodily needs, enzyme inhibitors can be used to halt it or slow it down. Enzyme inhibitors are often found in drugs like penicillin. Penicillin contains inhibitors which inhibit the enzyme that catalyzes the biosynthesis of bacterial cell walls. The reproducing bacteria will have faulty cell walls that burst.
Enzymes are affected by environmental changes. For example, 37 degrees Celsius is the optimum temperature for enzyme activity because that is the average temperature of the human body. When the surrounding temperature varies too far from 37 degrees Celsius, the active site of the enzyme will change in shape or the nature and won't be able to bind with the substrate anymore.
This also applies to the pH value of the environment. Each enzyme has a different optimum pH value, and its active site will denature and change in shape if the pH value of the surrounding varies too far from optimum pH value. For instance, amylase, which is secreted in the mouth at a surrounding of neutral pH, will denature when it travels down the stomach where it's acidic.
Enzymes do more than just assist in the absorption of nutrients in our body. They are biological washing powders with enzymes that help to break down stubborn stains. When clothes get stained with oil, also known as lipids, lipase, which are enzymes, will break down the lipids in the stain.
That's all from Just Minions. Thank you for watching.